All right, this is Kate Scow again, and this is going to be part 2 of Lecture 1, which is for Soil Microbiology, Soil Science 111. And we're going to talk about the types of interactions that are found in soil among different members of the soil food web. So first we need to define a few terms on the left-hand side. There's these different levels of organization of life forms in nature, and we start from the smallest scale of the individual, a cell or an organism, which would be like one bacterial cell. A population is a group, an assemblage of organisms that are of that same species, or let's say OTU. So multiple members of the same, sometimes it can be the same clone, or it can be a population that will have some variation within it. And then a community is a mixture of different populations that are organized into some kind of set of relationships. And then an ecosystem is a self-regulating biological community which consists of a number of communities and their physical environment. Some other important terms are that I think you've met up with in the lab are pure culture. Pure culture is a culture like a bacterial culture originating from just one organism, and it will represent one clone or strain, and everyone in it will be members of the same species. But really what's much more important in soil are consortia or mixed cultures, which are combinations of different species or strains of organisms that are often responsible for carrying out a lot of processes in soil. Pure culture sometimes can carry out important processes, but I think in nature you're often going to find things being, important functions being carried out by relationships of organisms. And succession then is a sequence of changes in populations, or it could be communities over time. And you might see this in decomposition of plant material or a dead vole, where you start out with an initial group of organisms, and in fact they may be shredders if you remember from the soil food web, and the first sort of guard of organisms, of microorganisms that are involved in decomposing often some of the more complex polymers making up plant material or the vole, and then they will create new chemical forms that then additional sets of organisms can carry out. And we see successions in both types of electron donors that are present, but also electron acceptors that are used. And we'll talk about this more in the class later on. Okay, so if we do a big overview of all the interactions that we can anticipate among populations making up the soil food web, we can look at these interactions in terms of the relationship between a first and a second population. So this is a very overly simplistic depiction of these relationships, but at least lets you get kind of an overview, a bigger picture of them. And I want to say that this kind of categorization, or cubby holes that we like to stuff biological interactions in, make these neat and clean definitions, this is a human problem. This isn't what nature necessarily wants. In nature there's going to be a lot of continuums and a lot of exceptions or deviations from these clear definitions that we like to impose and say this is how it works. So in some cases these definitions are not clear-cut, watertight. You may see some contradictions in some of the examples, but it's still useful to go through this exercise and get kind of a broader overview to organize what is a whole bunch of different kinds of interactions into some clear categories, because it really helps us to grasp the ideas and to think about Think about these interesting phenomena. So we have commensalism, synergism, mutualism, competition with antagonism as a component of that, amensalism, predation, and parasitism, with these different types of positive and negative or neutral, in the case of the dashes, impacts on the members involved in the relationship. And we're going to go through some examples of these different kinds of interactions. So starting with commensalism. Commensalism is a one-way positive exchange. One organism benefits from another's table scrap. You can see mens in here, which is, I think, Latin for table. So one organism may take the table scraps of another one. And this is a very common process that you see all the time in soil. It's very important for things to turn over in soil, and with pretty low specificity of the organisms involved. By that I mean it's not very clear, specific partnerships between this organism and that kind of organism. So it usually involves modification of the environment by one organism that in a way will benefit the other. But the first one may not benefit from it. So examples are metabolic products that are released, organic compounds that are byproducts when one population breaks down, for example, a complex compound like lignin that releases a variety of simpler chemicals that suddenly open up this organic material to a way larger group of organisms, because lignin degraders, they have certain talents. It's very difficult to degrade lignin. So not many can do that, but once they start to break it down, then there's going to be a variety of other things released that other organisms will use. That's one example. And another is when it's sort of more passive, if aerobic organisms jump in and consume all the oxygen in, say, a pocket of soil where there's a lot of organic carbon, then the environment is going to go anoxic. And suddenly anaerobic organisms, which didn't have a chance before, are going to have an opportunity to be engaged in breaking down that organic material. And also growth factors are specific organic compounds that are produced by that are important for organisms to carry out their processes. They're not necessarily nutrients. They're usually organic compounds. And these can be produced by one set of organisms and used by another. Synergism, okay, so also called proto-cooperation. This is a two-way positive exchange that's going to be beneficial to both organisms involved, but it's not necessarily obligatory. They don't necessarily have to do this to carry out their functions. And there's tons of examples of this. And actually, the fact that these proto-cooperation interactions are so common in the breakdown or the turnover of all sorts of material in soil, it makes it kind of complicated when we're so fixated, we're so obsessed as humans with pure cultures. We want to get that pure culture out that carries out an important function. But a lot of times pure cultures don't do these things, and you try to study like a process like methanogenesis, and you just keep trying to isolate a pure culture of one bacterium that carries this out, and you can't make it work because one organism doesn't carry it out. It's a consortium of organisms engaged in synergism. An example is in flooded soils, and we're going to go in more detail in this later, but bacteria that carry out fermentation are going to create byproducts, simple carbon compounds, and also hydrogen gas that then methanogens can put together to then create methane. And then those methanogens will reduce the toxicity that could be brought about by the acetate and hydrogen gas that's building up around the fermenters. So they're going to provide a service back by using up those products and then allow the fermenters to carry on with what they're doing. Another example is in pollutant degradation, where you have one organism that could convert a pollutant to a metabolite that actually is toxic to it. And this happens a lot, this kind of committing suicide by breaking something down. But if there's another organism present that can break down that metabolite, then it will reduce the toxicity to the organism that carried out the first step. And then together, those organisms can break down this material. That's another example. Okay, mutualism, also symbiosis. And I do realize that these terms are used in different ways by different people. This is just how we're defining these terms in this class, but you're going to find differences in other classes or in other books. Anyway, mutualism is, again, a two-way positive exchange between two populations, but we tend to look at this as more specific. This exchange allows a new biological trait to be expressed, and it could be it's often an obligate relationship. Like, without being in this relationship, these organisms will not be able to maybe survive, or more commonly may not be able to carry out this special process that they can carry out when they're living together in this association. And there's many examples that are so important in soil microbiology of this kind of interaction. And when we get deeper into the class, in the nitrogen lecture and in the rhizosphere lecture, we will look much more at the plant microbe symbioses. Rhizobian legumes, Frankian shrubs, mycorrhizal fungi, and almost every plant on Earth. We will look at examples of that and actually look at them in the lab. But we'll wait for those, but let's talk about another kind of symbiosis, which is lichens. And I love this photograph here because it shows an algal cell, this round thing that is embraced in this intimate arms of this fungus, which is the other member of the symbiotic relationship that is a lichen. So let's take a look at that. So lichens are an association between fungi and either green alga or more commonly cyanobacteria. Now remember, we already talked about cyanobacteria in the first part of this lecture, and they are important in desert systems. They're part of these cryptogamic crusts that are in deserts. The difference between green alga and cyanobacteria can also fix nitrogen. That's one difference between them, which you can see this is pretty amazing if you have a symbiosis with a fungus and then someone who can bring carbon in from the atmosphere and nitrogen in from the atmosphere. The benefits to the fungus of being in this relationship is they get carbon and nitrogen if a cyanobacteria is involved from this photoautotroph who's in the relationship. And then the photoautotroph, the alga, the cyanobacteria, is going to get protection from drying and grazing by being inside this fungal thallus. There's that word again, thallus, made up of all these mycelia bundled together. And they also are going to get minerals that the fungus will mine out of the rock. And also they're going to have a physical structure that lets them fix nitrogen, because nitrogen needs a specialized kind of environment that's protected from too much oxygen for it to take place. The fungal thallus helps provide that. And it's really interesting to look at this picture and see. Here's one kind of lichen shown to the left, and you can see this big fungal body here, this mass of mycelia with filaments throughout, and this kind of big physical thing that is basically what you see when you look at lichen. It's going to be primarily the fungal mycelia, and then you see this thin layer of the algae here that are protected in the way that we just described in looking at the benefits to the phototroph by being in this fungal thallus. And they can, they're protected from desiccation, predation perhaps, unless the predator eats fungus, and also in this environment where maybe the oxygen is less. So as we talked about before, These lichens and the cyanobacteria are a really important source of carbon and nitrogen in certain ecosystems, and not only deserts but tree canopies. Lichens living up in the tree tops can actually fix a bit of nitrogen, and it can drip down with rainfall, like in tropical rainforests, and be a source of some nitrogen to the ecosystem. Okay, competition. This is a more low specificity type of interaction, and we classify competition into two types: one exploitation and one interference. So exploitation is more like a resource. Okay, competition always has to be talked about with respect to a resource. Competition for what? That needs to be sort of designated here. And in this case, it might be a carbon source. And in exploitation, one organism is going to deplete that resource without physically restricting access to another organism, and therefore be able to outcompete them for the resource. And an example is like organisms with very fast growth rates, where they can just jump on, colonize, suck up all the resource before a slower organism can get a chance. And you definitely see this on Petri plates, where those organisms that are fastest are the ones that are going to grow those big honking colonies, and there's a whole bunch of other organisms that are lying on that plate who would be happy to grow, but there's no more resources left because the greedy ones got it first. And also another example is with production of compounds like siderophores, which are good for getting iron that some bacteria produce. We'll talk about this more. Or emulsifiers. Bacteria can produce emulsifying agents that are released around like oil spills to help create better access to breaking down those that particular insoluble kind of compound. Interference is the other kind, where you deny access to the resource by behavioral characteristics. And a good example is physical blocking by colonizing good spots, for example, roots, by trichoderma being a fungus that is not harmful to plants. It's actually somewhat beneficial. And in fact, this is used in biological control. Sometimes people will inoculate plants with trichoderma to get them to kind of coat the roots with their hyphae and prevent pathogens from being able to grab hold of things. So it's interesting to think about using these naturally occurring biological processes, and how can you perhaps tap into them for biological control. Amensalism, specificity in this is variable. That means that it's not necessarily a very strict relationship between narrow groups of organisms. It could be a broad process, or it could be more specific. And here, amensalism is where one organism is antagonistic to another, and often because it releases inhibitory compounds, chemicals, or changes the environment to make it more inhibitory to others. And an example could be lowering the pH by producing acidity. It could be by releasing antibiotics that might inhibit other organisms, or other kinds of toxic compounds. And there's been a lot of research in plant pathology trying to identify organisms that have some of these properties that they might be able to use and inoculate those organisms to shut out pathogens. An example is sulfur oxidizers. Sulfur oxidizers can lower the pH, and that can affect the growth of streptomyces around them. And this is can be used and is used sometimes as this kind of more general biocontrol, where you could put in elemental sulfur to the soil, and sulfur oxidizers are going to want to oxidize that elemental sulfur. That process will result in lowering the pH in general. And so if you have this sulfur applied around plant roots, then pathogenic streptomyces will have less of a chance of getting a hold, of being able to survive and create problems. And you can also see this kind of thing in Petri plates, where You can see a mensulism in a petri plate, where sometimes you'll find on a plate that's colonized by a lot of colonies, too numerous to count, but you'll find maybe one organism has this zone around it where it's all clear because it has produced an antibiotic or some toxic chemical just immediately around it that prevents other organisms from being able to grow there. And in fact, people use this sometimes to try to pick out organisms that might have antibiotics and to try to select for them and learn more about them. So again, petri plates are great places to see all kinds of interesting things. Okay, predation is a relationship between two organisms in which one engulfs or captures, digests the other. And usually the predator is big and the prey is small, as opposed to parasitism, which we'll talk about next. And we have lots of predators. You remember this from the first part of the lecture. We've got protozoa, we have nematodes, we have slime molds, and there's a number of others as well. But those are certainly important ones. And the impacts of predators are impacts on the community structure. They're going to selectively graze on some organisms and not others, so the community structure can change. It can increase the rate of succession in communities through their pressure on particular groups. It can increase rates of decomposition by rapidly turning over the population and keeping them sort of in the early stage where their rates may be higher. The decomposition, say, by bacteria or archaea of organic material. And also, predators are really important in releasing nutrients as waste products and helping recycle nutrients that are tied up in the microbial biomass. And we'll look at an example of this in a minute, but very important role of predators. And here's just another food web that shows some of these relationships of bacteria and fungi and their predators. It's just another depiction of a food web, just to remind you of these types of relationships. Okay, and parasitism is where one organism causes harm to another organism over a very prolonged period of time, and usually it's a smaller organism that's going to have an impact on a larger organism. And the parasite might require a host to survive, and therefore it's an obligate parasite, or it may be able to survive in the absence of a host, be a facultative organism. And viruses are good examples of what are often obligate parasites, and Dellavibrio is an example of a bacterium that can survive free in nature until they find who they're going to attack, and then they do this horrible thing. Here's this Dellavibrio drilling into a victim bacterium, taking over the inside, reorganizing things, reproducing, making lots of babies, and then the poor bacterium bursts open and all these terrible Dellavibrios come out again. Here's a nice microscope picture showing the Dellavibrio attacking these larger rods, and one coming in right there. A little bit of drama for you. And then this idea of liberating the nitrogen, I want to show you this in this kind of complicated, but I think it shows it pretty well, figure. So bacteria have a carbon to nitrogen ratio that is lower than protozoa do. And that carbon to nitrogen ratio is more like 5 instead of 10, which means that the percent of nitrogen in bacteria is higher than the percentage of nitrogen that is in protozoa. This means when protozoa eat nitrogen, they're going to get more nitrogen than they need. And that's shown here here's this carbon to nitrogen ratio of this bacterium. You can see for this many carbons, there's 2 ends. The protozoa only need 1 of those ends. So when it consumes that bacterium, it's going to excrete ammonium as a waste product. Now, adding to that is the fact that the protozoa is also going to respire. So some of that carbon is also going to blow off as CO2, which means that even more of the nitrogen is not going to be needed and will be released. And this is demonstrated to be an important source of nitrogen for plants to take up, is this liberation of extra nitrogen that the predator doesn't need through this phagocytosis process, this engulfing and digesting of the bacterium and release of ammonium. And this is a little study here that you can look at that shows in a very simple system that adding protozoa led to higher biomass of the plant, because the plant was able to get nitrogen from the protozoa breaking down the bacteria. Okay, and then just I want to remind you that genetic exchange is also going on between individuals, and it's often happening either through vectors, through, for example, transduction, which is, remember we talked about this with the viruses moving genetic material around. We also talked about plasmids and conjugation, which is the way in which plasmids are moved from one organism to another, and the genetic information is exchanged. We also talked about competence. Remember I talked about some bacteria dying and releasing their plasmids, or it could be other pieces of DNA, and some bacteria can take them up directly from the environment. And there's also transposons. There's all sorts of creepy stuff going on. But that's another important type of interaction, is this movement of genetic information among different groups of organisms. Okay, and then I'm done with this lecture for now. That's part 2. So now we've finished lecture 1. And I just wanted to leave you with this, just for your information, but a very nice picture that was put together by a student of mine a long time ago, showing the different major groups of organisms along the column headings, and then some different information along the rows. One thing that's interesting is scale. We go from 1 micron bacterium size up to centimeter size, and earthworms. The dot of an eye is a bacterium, then a roly-poly is an elephant, and an earthworm is 10 elephants. So enormous scale in sizes that we see among these different organisms. And it makes you think about like how a collembola is going to think about a bacterium. I mean, collembola is going to still be way too big to see a bacterium, whereas a protozoa is going to be able to see it with their eyes, if they had eyes. Here's some information on what organisms eat, the mode of living, and we talked a little bit about this, and we'll talk about it more later, but most smaller organisms, microorganisms up to nematodes, are aquatic organisms. They are aquatic, even though they're living in soil. So they are sort of stuck inside water films in the soil, and it's no problem when there's plenty of water in the soil, but when the soil starts to get really dry, these organisms get trapped in these water films. They can't move as quickly, and they're not going to have access to nutrients and water in other areas. The exception being, remember we talked about fungi. Fungi are able to send their hyphae across air-filled gaps to go tap into maybe nutrients or water in another little pocket across the way. These higher organisms, collembola, mites, isopods, earthworms, they've broken free of the water film. They are like us. They contain their water inside themselves, right? How clever we are that we're not doomed to be aquatic organisms. So they have some other kinds of abilities to move around much more in soil, and perhaps leave or go deeper in the soil when things get really dry, although they'll also be vulnerable to desiccation. And biomass, let's look at the biomass. Most of the biomass is in bacteria and fungi, way more biomass than nematodes. Don't let excitable nematode people tell you that the biomass is so huge in soil. It's big, but it's nothing like the bacteria and the fungi. And archaea, by the way, they're not there, you notice. Even earthworms, I mean, they can get, with a lot of worms, you can get a pretty high biomass of them, but in general, most of the biomass is in these smaller organisms. And then the other thing is the generation time. how fast it takes for a new generation to appear, or how long organisms live. They turn over much more rapidly. The bacteria and the fungi do, particularly the bacteria, and then we get into much longer times in the higher organisms, meaning that there can be tons of rapid change and response and evolution in microorganisms, which we know very well that this happens. And they can respond quickly to changes and sometimes offer up different sort of choices and opportunities. And are going to be doing things a lot faster than their predators or other organisms. Okay, so with this, I stop this lecture and see you in the next one.